This is Mrs. Holmer. I'm the biology teacher on Team 4, and today we're going to look quickly at DNA structure and replication. Now, DNA, by now you should know, is the molecule that stores and transmits all of our genetic information, and it's also that recipe book by which proteins are made, so it's involved in protein synthesis. So we've been looking at DNA all year long. Now, DNA is a nucleic acid, which means it's composed of nucleotides. Here's a sample of a nucleotide here. Nucleotides have the structure of a phosphate group, a sugar, in this case deoxyribose sugar, and a nitrogen base. And the nitrogen bases we use for DNA are A, G, C, and T. And we take those nucleotides, which I have one circled here, and we're going to link them to each other in two strands for DNA. And we're going to link them through a covalent bond between their sugar and their phosphate. So you can notice all the way down we have a covalent bond between sugar and phosphate. And this is true on both sides of DNA. This is why we say DNA has a sugar-phosphate backbone, because the sides of it are composed of the sugars and phosphates linked together. Now in the center, we have our nitrogen bases, C with G, and down here, A with T. So our nitrogen bases are complementary. C always pairs with G, A always pairs with T. How do you remember that? Well, apples grow in trees, cars go in garages. So hopefully that will help you remember, because it's possible on the air test you would have a sequence of DNA and you would have to give the complementary strand. And so you always pair the nucleotides up with their complementary bases. Lastly, they're linked together in the middle here with a hydrogen bond, and that's important because hydrogen bonds are very weak, which means they can be broken very easily for things like replication, which we're going to look at in a minute. Just a couple quick things about the structure of DNA. DNA is considered anti-parallel, which means it goes in opposite directions. You can see that on one side, these nucleotides had to be flipped upside down so they would fit properly, and that's called anti-parallel. And finally, DNA is not just a ladder, it's a ladder that's been twisted up, so it's called a double helix. You can see there that kind of twisted shape of DNA. So again, here's our structure of DNA. Here's a little simplified structure. Looks like that ladder with the sugar and phosphate on the side and the nitrogen bases in the middle. And then finally, we have our double helix. This is when it's been twisted up in its normal structure. So now that we've reviewed the structure of DNA, we're going to look at DNA replication. This is when we take one strand of DNA and we make a copy of it. So we end up with two strands of DNA. This is going to always happen before mitosis and meiosis, so we have an extra set of DNA for our new cells. Now the steps of replication are fairly simple. We take our DNA, and the first thing we have to do is we have to open it up. And then we're going to use either side of the DNA as a sort of template. And we're going to just build in the complementary base pairs. So you see we started with an A, we're complementing that with a T. And we're going to do that on both sides so that when we're done, we'll have two completed strands of DNA. So just a little bit more detail. Here we are in the beginning. We have our DNA. We're going to open that up again. Now remember, that's done by an enzyme. The enzyme that does that is called helicase. Enzymes are part of a lot of processes in our body, and you can tell it's an enzyme when it ends in ASE. So now that I have my DNA open, again, I'm going to use either side as a template. Here's another picture here where I have this top strand called the leading strand, and I'm using it as a template to build this new strand in. And then I have the bottom strand called my lagging strand, and I'm also using it as a template to build my DNA in. I'm building those both sides. And then finally, I'm just going to twist them up. So once I have that new strand built, I'm going to twist it up on either side. And my final product is going to be two completed DNA strands. Now, if you look closely, you'll see here this original DNA was purple, and the new DNA strand is red. So we have half of our new DNA is old and half of our new DNA is new. And because we keep 50% of the old DNA in every strand that we create, we call this semi-conservative. So the process of DNA replication is semi-conservative. That's all I got. Good luck on the air test.